Yeah, what I'll really be emphasizing today is sort of the continued advancements in spatial biology. I introduce a couple of new classes of experiments that we're doing now, but there is one key theme: how to obtain a maximal amount of information, multiple layers of information on the same single tissue section slide. This is a great example. This is an excisional breast cancer biopsy imaged at the whole transcriptome level, 19,000 different transcripts. At the same time, 76 proteins, all simultaneous on a single slide. Not shown is a aligned H&E image associated with the same patient sample. So from a single 5-micron section, you're imaging the whole transcriptome, 76 proteins, and an H&E aligned image from one 5-micron hospital grade sample. This is really where I was hoping spatial biology could get to 7 years ago when I first introduced the first platform, and now we're starting to get there. Let's see if I can move in the right forward direction. I'm going to be mainly at this seminar. I'm mainly going to be talking about the genomics DSP, the cosmics SMI, and how it interacts downstream with the cellscape system. And I'm going to give you just a little preview of a brand new platform that's going to come out this year. It's called Paintscape, and it sort of is imaging. The other thing that we don't talk about too much is high resolution DNA imaging. So let's get started. I'm going to go really fast over this section. This is not a launched platform yet, but it will be this year. And it's sort of the neglected aspect of spatial biology, if you will. This is going to be high resolution imaging of the three-dimensional chromosome architecture in single nuclei. And it looks like this. It's a blow up of a single DAPI nucleus, single nucleus of a single cell. And we're imaging our way through all 23 chromosomes, figuring out exactly where everything is at. And at the same time, we see where it's at. We actually look for things that are like loops and tads associated with the structure of the nuclear structure, because I always say it all starts here. It starts in the nucleus. Something up there is opening up an area that gets transcribed, that gets translated, that makes protein PTMs, but it all starts up here. And there is no other platform now that can currently image in full 3D resolution the nuclear structure, and that's what we'll be launching this year. It's called Paintscape. I'm only going to highlight one aspect of this, especially if you're a cancer researcher. There's a brand new area of biology that's opened up. It's called extrachromosomal DNA, ecDNA. And it turns out in cancer, driver mutations tend to accumulate in the ecDNA, and you can see the ecDNA trace on this spatial diagram of all the chromosomes in a cell, and you see that long line up there. That's an ecDNA in the nucleus interacting with actually all 23 different chromosomes. So, an exciting area. Just keep your eyes open for it. You may see it. You'll see it later on this year. These 4 areas are the main things I'm really going to be emphasizing this year. Okay. So one of the key things is that you're going to see how important the protein layer of information is for, I think, all experimentation. But especially you think that, okay, once you're imaging the whole transcriptome, what else is there? And I'll show you how important the protein layer of information is. I'll describe some of the recent comparison papers that are coming out. I'll also be describing for the first time ever, the data analysis sometimes is a blocking aspect, how much information you can get over a given period of time in spatial biology. And we developed a large language model method of interacting with this high-plex data. You can go home on your own little laptop, have a glass of wine, and actually have a conversational analysis with your high-plex data, and I'll show you how that works. This is now fully commercial. And people always ask me, how long is it going to take to get into the clinic? The clinic's here now. I'm going to show you what a patient report looks like coming out of the geomics inside a diagnostics laboratory, just to give you a feeling for where this thing's going. And then I'm going to introduce what I think is a whole new class of experimentation. And this is where we combine live cell biology functional measurements with high resolution spatial biology. And we bring these two fields together to really enable spatial biology in a brand new way. So those are the four areas we're going to go over. It's always worth remembering how young this field is. 7 years, the whole field is 7 years old. So even if you didn't do a single experiment up to now, you're only 7 years behind. And most people are, brand new to this field, which is good. When I put together, I had the first idea, that's back in January of 2015. I was working with MD Anderson Cancer Center, and I was watching how they tried to understand their immunotherapy clinical trials. And I was thinking they're never going to understand the clinical trials with a 4-color microscope, just never going to do it. So that's when I had an idea on how to do unlimited plex microscopy. That was January of 2015. I scribbled it down on a little tiny paper, waiting in the hotel lobby in Houston, Texas to give a talk at MD Anderson. And it turned out to work. Now, everything we did on that little scrap of paper actually did work. And that's how we launched the geomics in 2019. And that was 84 genes in space. And when I did that, people said it's impossible to do 84 fish experiments at once because everybody had done them 1, 2, 3, or 4. But we launched with 84. But I didn't build that geomics instrument to do 84 fish experiments. I did it to do 20,000 fish experiments. So with the same box, within 2 years, you were doing whole transcriptome fish. Roughly 20,000 genes. And as we were doing that, I was building an imager, a similar thing with the imager. We launched it in 1,000 genes, which is about 5 to 10 times more than anybody had ever done. And within 2 years, we are now imaging the whole transcriptome. So you didn't have to buy a new box. I mean, what's important is when you're building a platform, you got to think about the chemistry. The chemistry is the beginning of a platform. You figure out your chemistry and you build a platform around it. So when I figured out the chemistry that I was going to use, the hybridization chemistry I use, I knew I didn't have to build a whole bunch of new boxes every time I wanted to go higher in plex because the chemistry intrinsically scaled to the plexity I wanted. So no new boxes. We just let the chemistry advance inside a platform so you don't have to throw any instruments away. After a couple of years, they're all there. They're ready to go. This is where we're at, whole transcriptome. We're now up to 1,200 antibodies at a time. So I'll show you what that looks like. And when you're at 1,200 antibodies, I got 130 pure post-translational modifications I look at also in space. And I'll show you how meaningful that is. Okay, so these are the platforms I'm responsible for as CSO of Broker Spatial Biology. They're all intrinsically multiomic. So you can see that from day one. That's the Multiomic. We just made our diagnostic platform called the Encounter. It's now Multiomic. There's Cosmics at 19,076 plex on protein. There's Geomics at 18,000 and 1,200 plex antibodies. And here's a reminder of the chemistry. So this is the most important decision that you ever make when you're building a platform. It's what you do for the chemistry. The rest is in the noise. Okay, so you can see what it is on the left-hand side. When I first got to NanoString back in 2012, one of the first things I did was ran a huge clinical trial program in breast cancer. We did over 2,000 breast cancer patients using that chemistry down on the bottom. And those FFP samples spanned 35 years. And when I looked at those 2,000 samples, we had success with about 98.5% of those samples went through. I got data from every one of those, all the way back to 35-year-old samples. And that convinced me that a hybridization-based approach for archival FFP is the key enabling thing I wanted to accomplish. So every design decision I made on the instrument was around one thing. Could it be best in class on hospital, real world hospital FFP. That's why everything we do is hybridization. It's all in a very small footprint, 35 to 50 nucleotides big. That's how many nucleotides it takes to resolve a whole transcriptome. That's why I picked 35 to 50 nucleotides. There's a reason for that. And we made it multiomic from the very beginning because we knew how important multiomic was going to be. I didn't tag proteins on at the end. Proteins have been with me from the beginning. Okay, so let's look at what's new on the cosmic side. And we've been imaging the whole transcriptome for about 2.5 years now, which means I've been imaging in R&D the whole transcriptome for about 6 years. These data sets are now all public domain, so you can actually go out there and see exactly what's happening in all those different organs. Totally public. We're averaging over 2,000 transcripts per cell and over 900 unique genes per cell. We also put another one out in the public domain. It's the largest multi-omic data set ever collected on planet Earth. And that's the breast cancer sample you see over there, imaged at the whole transcriptome. It's got 64 proteins layered on it, and that's also in the public domain. There is no multi-omic data set of this size and scale anywhere else on Earth except where we put it out there. Lots of information in that data set. I sort of follow where our first papers come. It's very nice. The whole transcriptome, very first paper, it's in the Journal of Clinical Investigation. That's what we built this thing for, clinical translatable results. And it turned out that we discovered a whole new mechanism by which B-cell lymphomas hide in the germinal centers of your body as they multiply. And whole transcriptome was able to find a brand new mechanism. They did that. It's called DNA damage repair, a special molecule called ATR. No one expected it to be active there in that particular lymphoma, but that's the advantage of a whole transcriptome. I discover what biology is really doing in there. I don't have to read it in a paper and see if I find it in the tissue. You discover it in the tissue. The first time I gave a talk on whole transcriptome, actually, I got a call from a drug company called Merck, literally the day after I presented up in Boston, Massachusetts. And they said, I want to start looking at that. I want to image a whole transcriptome. I told them, it's not going to be commercial for about another 6 to 9 months. And they said, I don't care. Give me whatever you got in the lab and let's start working together. So we did that. And one of the projects we did with Merck, we've gone through about 8 different indications now, but this is one that I can talk about. We also were the very first persons to do CRISPR-Cas9 editing in organoids with Merck. And it's multi-omic, so it's whole transcriptome readout. It's about 75 proteins, all on organoids that we grow, slice them into 5-micron sections. It's the only multi-omic CRISPR screen that's ever been accomplished. And I recognize, take a look at that paper there. It's and if you have an interest in CRISPR-Cas9 things, send me a note. It's a very exciting part of research where you're proactive in what's going on. We were engineering the cancer-associated fibroblasts and watching how that affected primary tumor growth in an organoid. Exciting work. Okay, this is probably the only thing I'm going to talk about comparison-wise, but it's an important new paper out in Nature Communications, April of this year. It's the first time a clinician ever did a cosmics versus xenium comparison study. I think it's important because that's what we built this instrument for. It's for clinical translational work. So this is a clinician, and he did it in a very exacting way, as a clinician does. It's totally a controlled study. So they have over 100 tissue sections. It goes into one person that are experts in actually sectioning. Okay, same person. At the exact same time, they put those slices into a xenium, and the xenium is running in top flight. 10x people are all over that box the whole time. Same time it goes into cosmics, they're run. And they really look at, is it suitable to do a full-blown clinical cohort? That was what they wanted to know, because that's what that investigator had to make a decision for inflammatory bowel disease in the United States. This is the major consortium, and he was responsible for making that decision. And you can read that paper, and on every every aspect, every important clinical attribute associated with how they were evaluating that disease, yeah, Cosmics came out superior. And I think, one of the key areas, the reason why, especially look at item 3. This is the most important thing. It all goes back to the chemistry. Okay. Hybridization chemistry is invariant on whatever atmosphere the tissue is in. That's the way hybridization works. But if you're doing something like rolling circle amplification, what did they find in rolling circle amplification is that when they hit inflamed regions of the tissue, the quality of the signals associated with the rolling circle amplification goes down, and it goes down significantly. So the very thing they were trying to study, which is early inflammation in inflammatory bowel disease, was invisible. So the most important biomarker they needed to find showed up in the Cosmics. It was absolutely invisible in the xenium. So they called up the 10X people and said, why didn't you find it? Where did it go? And then the xenium people, the 10X people said, well, you know what? Maybe if you turn the QV value filter for our report out, turn it all the way down, maybe it'll show up down there. And lo and behold, it did show up down there. The thing that we saw in Cosmics is in there. But the fact is, the rolling circle in the inflamed region, the quality of those transcripts went way down underneath their filter. So if that clinician had not been doing the Cosmics, they would have missed their most important biomarker because it fell underneath the QV value because the rolling circle is not invariant as a function of the tissue. So that's the main bottom line there. If you're interested in translating things to the clinic, you got to look at the chemistry again. Can the chemistry translate? And if you actually look at rolling circle, there's not a single diagnostic made on planet Earth using a rolling circle method. Zero. Hybridization, there's a plenty of them, hybridization clinical assays, but there's not a single rolling circle. So anyway, that was that clinician. That was the discoveries they made. Okay, these are some additional ones that are coming out. Large patient cohorts, over 500 patient non-small cell lung cancer, a complete whole transcriptome study. And another thing to realize there, these were at 2 different hospitals, one in Australia with Arutha Kulasinghe and one at Yale. And another thing about hybridization is that it's invariant from the site. So they were able to harmonize the data from Yale with the Queensland data perfectly. Harmonized, no problem. And you can see it right there in the middle. Complete harmonization, single cell RNA-seq level transcripts, and keep an eye out for that. This will be out in Cell. Okay, some people always ask me, why do I care so much about hyplexity? And I think that one in the middle is one of the key reasons. First time I looked at pancreatic cancer at the whole transcriptome level, what did we find? What we found is one of the largest regulatory mechanisms in pancreatic cancer is developmental nerve biology signals. And everybody says, yeah, nobody's looking at nerve cell biology with their little panels because, yeah, they're not paying attention to that. It's one of the biggest signals. It's the nerve cells actually talking to both the immune cells and the tumor cells, and the stromal of PDAC is one of the largest signals you'll see. But it was totally invisible. Everybody was looking at with these little tiny panels. Nobody was paying attention to the nerves, but it's right there. What's neat is I was just maybe a month ago, I was at AACR, and actually somebody measured using nerve traces right from the back of the spine into the pancreas on PDAC, and it's totally crazy. The nerve system is haywire associated with pancreatic cancer, but no one had actually ever done it before. We found it here molecularly. Now they found it imaging in humans. So it's real, and it's a big term. And it just sort of reaffirms to me, every time I look in tissue, I see something that is unexpected. Okay, that's why you can't be looking at these little panels. You have to see all the biology. Okay, some people think it's harder to analyze when you have more data. And I think it's totally the opposite direction. Because when I have limited data, what do I have to do? I have to infer the biology off of my little cell types. That's why people are so crazy. I got to get my cell types right because that's all the information they have. And they have to infer the biology from the cell types. When you measure at high plex, there's nothing to infer. You just measure the biology. I don't even know that. I don't even need the cell types to tell you what biology is happening in that tissue. I just plot out the pathways. There's a breast cancer biopsy, a single one. There's 2,751 reactome pathways that I've just measured. Take your pick. There's MAP kinase, there's hypoxia, TNF alpha. You're not having to struggle with cell typing to do that. You just put the biology right on the tissue and you measured it. I didn't have to infer it. No fancy bioinformatics going on here. I don't have to infer that the M2 macrophages are doing this or that. You just measure what the biology is. And you say, well, I got the whole transcriptome. What do I need protein for? And hopefully I'll convince you here with a couple of examples how important that is. One are the obvious ones. So like that second panel to your left, that is all the mRNA associated with MHT class 2 antigen presentation. But then look at, I got HLA-DR right next to it. And you can see cell by cell, that protein image matches what the mRNA pathway is information. But then many times you see something that's not just, it can be contradictory, but oftentimes confirmatory of interesting biology. And I'm going to talk to you about that. So this is the foundational data set. I think I've talked about this, but this is where every time I measure, this is what I got. So we continue to push. We've always been known as being the best segmenters in the world on these things. We've now launched the world's only three-dimensional segmenter for hospital-grade FFP, 5 and 10 microns. You think it's so slim it doesn't matter, but there's a lot of overlapping cells, even in a 5-micron excisional biopsy, and now we can resolve that. Only group in the world commercially that's doing this now, but it's now in atomics. So yeah, a good advancement. And when you go multi-omics, you really got to work on the analytics for multi-omics. People talk about multi-omics data all the time, but do they talk about the analysis? Okay, this is one of the first analyses that we wrote where you take the high-plex transcriptome data, high-plex protein data, and then you do simultaneous analysis to do fine granular subtyping, cell typing. So it's the protein and the RNA information are both battling it out. The algorithm figure which one has more information content than the other, and it makes a call. It's the best calls you can make in cell typing at the current time. And every time we do something like that, it's open source. So you can download it, look at every equation I use, look at how we do it. It's yours to examine. If you don't like my equations, just change them. But it's all there in the open. We don't do anything where it's hid. And this is a very useful place to go. All sorts of tricks, everything that comes out of my lab from the spatial analytic groups, I put this thing out there almost immediately. So if you actually keep your eyes on what's happening in Scratchbase, you'll be up to date to about 1 month on every state-of-the-art activity we're doing. Don't wait for the papers to come out. Too slow. Okay. Now I'm going to tell you about a brand new class of experiment in oncology. I think it has applications everywhere, but I'm going to show you what it looks like to do it in oncology. I'm going to combine the live cell platform called the Beechem Discovery inside Bruker Spatial Biology with high-resolution spatial imaging to do something that's never been done before. Okay, so I'm interacting directly with surgeons now. I used to always spend all my time interacting with pathologists, but once I thought of doing this live cell combination, I'm interacting with the surgeons in the surgical room. So what am I doing? We're doing head and neck cancer. So I'm interacting with the surgeons. They take the head and neck cancer out. They take half of the sample. We're going to dissociate it and do live cell biology on them. And I'm going to try to determine which T cells in that tumor or that biopsy know how to kill the primary tumor cell. Okay, that's what I'm going to do there. I take the other half of the biopsy and I do high resolution whole transcriptome imaging with a special spike in that allows me to match it against a T cell repertoire RNA-seq experiment. So molecularly, I can find the unique T cells that know how to kill a tumor cell. I think it's easier to just see the movie than to listen to all my words. This is how it works. So what you're looking at in the upper left-hand corner is a little cell culture area. It's on a semiconductor chip about this big, okay? And he houses anywhere from 5,000 single cells out to about 22,000 single cells. So right in the operating room, those things go out, we dissociate them, and with light, I can grab a T cell and put it in a little culture well. That's culture well 1584. I can grab a tumor cell and put it in there with it. And I take a movie and I say, does that T cell know how to kill that tumor cell? So when you're looking at that big fat tumor cell up there, the minute you see it go yellow, you see that thing go yellow? That means the T cell killed it. I got a caspase assay running in that little culture well, and I know exactly what it's did. I'm also measuring interferon gamma and a whole handful of other cytokines and chemokines. So I unequivocally know those T cells in there with that tumor cell know how to kill. I take a little light beam and I pull those T cells out, and then I'm going to RNA-seq them and do T cell repertoire sequencing on them. So I know exactly the variable domain, the CDR3, that actually knows how to kill a primary tumor cell. Okay, then I got the other half of that head and neck cancer down there. That's the spatial one. And I'm going to find the T cell that matches the molecular fingerprint of the T cell repertoire, and I'm going to tell you where they are. Okay, so let's do it on a couple of patients, and I just let you see what it looks like. There's patient 137 on one side, patient 120 on the other. Patient 120's got 32,000 T cells. Patient 137's only got 6,500. So if you're just looking at that thing, you say, well, the one on the right probably going to respond to immunotherapy. The one on the left, poor person doesn't have any T cells hardly at all. But when we made the functional measurements on those two things, what did we find? That patient over there with 32,000 T cells didn't have any T cells that knew how to kill the primary tumor. Okay. But the one down here, 6,500, did. Okay, so why didn't that patient have any T cells that knew how to kill? So I'm imaging, and that's a tumor nest you're looking at. I'm blowing up one of the tumor nests, and then the stromal region's there, and you can hardly find any T cells inside the tumor nest. Yeah, it's like, I don't know exactly why. But then when you look at the multiomic version of that thing, look what you find when you do the protein overlay on this thing. You see that? See that red signal that's wrapping around the entire tumor nest? That is smooth muscle actin. That's a protein that's been laid down and is acting like a guardian around the entire tumor nest. So I could have stared at my RNA forever and never really know why they're not in there. But when you look at that protein layer, it's unequivocal. There's a protein barrier around that entire tumor nest. That's one of the reasons why protein's so important to have on the same slide. Okay, let's keep up with the logic. There's a blow up of that SMA wrapping around that tumor nest. And then there's all sorts of supporting data from the pathways. I can look at the pathways associated with extracellular matrix re-architecting right around that nest, and it's hot. And that's the RNA pathways that lead up to SMA around the border. Okay. So this is real additional information that I feel is essential if you're trying to understand whether that patient's going to respond or not. If I didn't have that information, I wouldn't know. Okay, so let's take a look at the one that did have T cells that knew how to kill. How many are there and where are they? So we did that. There's 6,500 of them. You can see all the different classifications. You know how many could actually kill? 33. There's 33 T cells in there that really knew how to kill the primary tumor. That was it. Okay. And this is sort of in general. You look at a bunch of T cells on any kind of excisional biopsy, you know what? If you think they're all killers, you're wrong. Even the CD8 T cells are not all killers. They may not have an antigen-specific killing mechanism. Okay, but we found them. I know exactly which ones killed because I measured it. There's 33 of them. Okay, and why couldn't they kill? Why does that patient still die? And that patient did die. And I'm walking through there and I find one of those killer T cells. It's not in a nest. And when I look at the protein around it, it's surrounded by cancer-associated fibroblasts. And those cancer-associated fibroblasts have made a prisoner of that killer T cells. Single cell prison of fibronectin is wrapped around that entire T cell. That's that glowing yellow you see there. Okay. So sometimes the protein barriers are like the Great Wall of China or walking around the entire nest. And sometimes they're isolated prisons holding them back. So that's why I think proteins are pretty important to have on the same slide. Okay. It doesn't always, okay, so this is a summary of everything I said. But I think you got the message. Oh, I didn't stop there. Okay. So that's a great thing, live cell biology. I'm also interacting with a group of surgeons that are actually in India. They're doing head and neck cancer also. It's a partnership I do with a company called Farcast. And the surgeons, when they're taking the material out, they take a microtome, a gentle one, cut a 500 micron thick living tissue section out of a patient. We drop it into cell culture to keep that thing alive. And then I'm going to apply the drugs ex vivo to the living tissue. Okay, so that's what it looks like. And then I'm going to image it with Cosmics. And these are the kinds of things you see. There's the head and neck cancers. And the ones in the upper right-hand corner means they don't respond to any kind of drug treatment. And those tend to look like that. And then you get another group that's down here that actually do have various complex responses to the drug treatment. And I'll show you what a blow up of those. And the non-responders are those ones that have tumor nests that are all blockaded away from the stroma, just like we saw in the other study. And then these ones over here are mixtures. And I think, the most important thing I'm talking about, this is a new kind of measurement. So we're doing live T cell biology, we're doing live tissue biology and combining it with high resolution spatial. And we're interacting directly with the surgeons. These head and neck cancer patients got about 9 months to live, 12 months. I can give that surgeon all this information in less than a month. And we're working hard to figure out how they can actually change their practice to actually give some of these patients a chance. Okay. And I'm going to go fast through this. This is what the barriers look like in non-small cell lung cancer. Sometimes they don't look like little fine lines. Here's a barrier. It's a extracellular fibronectin barrier around that protein nest, that tumor nest in the upper left. 150 microns away, the other tumor nest has got a different protein barrier around it. That's actually smooth muscle actin. I have no idea why that particular patient has got that particular characteristics, but it's there. Again, the protein layer is important. Okay, what are some of the other new things we're doing? We're imaging now the whole transcriptome of the mouse. Mouse is 22,000 genes you need to image. I wish I could tell you it was really hard to do that. It's trivial to do that. Hybridization scales, I don't really care what the plexity is. It just turned out a mouse was 22,000. If it was 40,000, I'd have done 40,000. It doesn't really matter. Hybridization scales, and there's the mouse. Upper right is the T cell receptor spike in that I'm talking. That'll be released this year. And if you really have an interest in that, send me a note. I can send material from my lab, and the people down here know how to charge for it and things. And then the lower right-hand side is something nobody talks about at all. Everybody talks about all these fancy things. What about the microRNAs? MicroRNAs are a really important RNA component that actually regulates, but no one talks about them because they're really hard to measure. There are 22 base pairs, and you have to have single base pair resolution. So we got it. We took our nCounter microRNA assay, where I'd already developed with single base pair resolution, and just ported it into space. And this will be the first time anybody's looking at spatial microRNAs ever. That'll probably be out in the early part of 2027. This is what whole transcriptome in a mouse looks like. 3,000 transcripts per cell, a couple of thousand unique genes per cell. 1,256 completely resolved reactome pathways over a 4-day gestation period in a mouse. Yeah, lots of information there. This is what the microRNA looks like at high resolution. We just did a whole poster on this, but if you have an interest, send me a note. There's some really fascinating biology here. It's absolutely brand new. No one knows what the spatial microRNA is happening. I can tell you one anecdote. It's kind of a fun one. I'm looking at macrophages in a placenta. There's a mom side to a placenta and there's a baby side to the placenta. And I'm looking at the microRNA regulation in a macrophage. It comes right up to the mom side. And I move a couple hundred microns over to the placenta side, and the whole regulation changes. Okay. Is that like a timer saying that I got so much time left, or is that nope? We doing all right, Priya, on time? Okay. Okay, this is a fun one. This is the first time this one's ever been done either. There's nothing more frustrating for me than having the person who spent all that money to put their precious disease sample in to the instrument, and it may be a heart or it may be a kidney, maybe a liver, lymph node, and then you hand it over to your analytics people. And I'm handing it over to an analytics people that may not know a lymph node from a germinal center or from a kidney or a liver. They're not tissue experts. They're just, they know they know how numbers work. But how do you expect to get brand new insights from somebody that doesn't even know a liver from a stomach or a kidney? So we wanted to give the data analysis back to the person that's an expert. So what we do is we take this high dimensional data and I put it into a data cube. I put it into a finite data cube that has all the domains, that has the Leiden clusters and a whole bunch of README for large language models so that they know exactly how to interpret what I'm giving them. And I hand that right to you. Works on your laptop. You can go home, drink whatever we were drinking last night. You know, where you drop, what is it, soju into a beer or something like that. You can just sit there and you have a conversation with the data. Okay? The conversation, take a look at the movie. I'll show you what it looks like. Let's start this thing up. And these different colors you see are different spatial domains in that tissue. And they're just defined by, they're just defined by the biology changes in the transcript. You don't know what they are. So I can bundle those things up and you can just ask a simple question. What is that blue domain doing? You know, what happens when the T cells get close to that domain 4 region? Is that what domain is that? Oh, and it may say that's the invasive margin of the tumor. Well, that's a pretty useful thing to know. And you just walk through your data like this, having a conversation. You're not writing any code. You are just talking to the data. And this thing works. I remember the first time I got an email back from a organ transplantation specialist, kidney guy named Bob Colvin at Harvard. He says, I'm a 75-year-old pathologist. I've never analyzed data in my life. And I'm sitting here talking to this kidney data and I'm getting the results. The happiest guy under the sun. It was interesting. I just got back from China and there's a group at Hong Kong told me the same thing. It says, all of a sudden, I can analyze my own data. Yeah. So I'm excited about that. Oh, I know what happened to the screen. So that's a fun thing. And it's active now. You can do it now. It's the only place in the world I know that you can actually get your high dimensional data packaged up and you can have a conversation analysis with it. Now, one of the things that's happening with CosMx data around the world is building foundational models. Okay. So I interacted with this pharmaceutical company from day one. A guy named Ron Alpha he's forming it. And he was forming this company with spatial biology was the discovery mode. It's a company called Noetic. And they built a foundational model that would predict the behavior of a cell just by pushing it anywhere in your tissue you want. And that's a foundational model built on CosMx 1000-plex. A drug company called GSK bought that in January for $55 million to be able to utilize that model that he put together with the CosMx data. Okay. Now, Ron wasn't done at 1000-plex. I knew that. And we're now doing 1 billion single cells at whole transcriptome using the CosMx whole transcriptome. And we're building foundational models around that. Okay. And this one has already just been put out. So it's not something happening in the future. There's a model right now called Taria, and Taria 2 is built on the foundational models around CosMx whole transcriptome that will predict the behavior of FFP slides just from the H&E image. Okay? You can read about it. You can read about the accuracy, but these things are going to happen, and they're happening now. They're happening fast. Yeah, hope you guys don't wait around for another 6 or 12 months to get enough whole transcriptome data to be a participant, because this is going to be happening. My guess is in 12 months, probably most of the foundational models maybe already be built. Okay, that was a pretty long tour through cosmics. Let's switch gears to the protein side of the equation, okay? The best protein platform, if you want to do high-plex protein discovery, is the genomics. There's nothing even close to it. 1,200 antibodies at a time, whole transcriptome, all on a single slide. And you look at tumor niches, tissue niches. These are very, very important. If people think that the only thing that's important in a excisional biopsy is a single cell thing, they're wrong. The biology happens as groups of cells work in these unique niches in tissue. That's the way tissue works. There is no single cell ruling how an organ responds. Groups of cells and niches is how organs work. So let's take a look at how this kind of experiment gets done. So there's a colorectal cancer tumor we're going to look at. It's got a little piece of normal tissue at the bottom. That often happens in CRC. And you see these three different tumor domains. So I'm going to look at whole transcriptomes, what's happening in the tumor cells up there in domain 4, what's happening to 1,200 antibodies in those tumor cells in domain 4, and I'm going to compare it to the same measurement of the normal tissue. Simple, okay? I can just do a simple volcano plot, and here's the pathway information coming out of the whole transcriptome. That's what's up in that domain 4 of the tumor versus the normal. All the pathways are apparent. Now I got 1,200 antibodies on the same region. So now my volcano plots all become combination plots between the whole transcriptome and 1,200 antibodies. All the little diamonds are all the whole transcriptome side, and all the dots are your volcano plots from your protein. And you really learn some unique things. TP protein P53 is up in a whole bunch of cancers, as everybody knows, and I see it is up in domain 4 of this particular colorectal cancer, and that's an indicator of stress associated with DNA damage. Okay. But look at the RNA as that triangle with that big circle up there, and look at the protein down there at the bottom. The really protein didn't change at all. Okay. The mRNA is way up there. Protein's just sitting there. What happened to all the protein? The protein is being post-translationally modified. So you said acetylation of P53 at lysine 373, another phosphorylation at serine 312. You get an interesting ATR phosphorylation, the serine 428. Those are all key indicators of the disease side. That's the domain 4 of the tumor. What this tells you is that the stresses of DNA damage repair on domain 4 of that tumor is totally linked to the single-stranded breaks in that. Single-stranded breaks, not the double-stranded breaks. And it's all being modulated by the post-translational aspects of those signaling cascade. Totally invisible without the 1,200 antibodies. I like this one a lot. It's the same colorectal, same domain 4 of the tumor. And I get this huge RNA signal for RPS6. That's a classic mTOR inhibitor marker. So you would say that, okay, that tumor is escaping. It's maintaining itself due to mTOR inhibition. You'd be absolutely wrong. If you gave that patient an mTOR inhibitor, it would do nothing to that particular tumor, even with the RNA way up there, because all the post-translational modifications that are happening in that tumor actually turn off that whole mTOR thing. Okay. So that's a good example of the protein layer really providing important insights that would even be related to drug treatment in that particular case. And I'll skip this one. Sometimes you get RNAs that are going in 2 directions at once. And just let the protein signaling answer which way it really matters. But anyway, hopefully you get a feeling that when you get to proteins at that level, you can really start having an interesting conversation with your data. Okay. And I'm going to end on this one characteristics that we've been working hard on is that I wanted to be able to do single slide measurements on any of these platforms up here and pull them out of one instrument and into another and combine them. So the first one I wanted to combine was actually the most powerful proteomic thing, 1,200 antibodies. I'm going to run it into geomics. I'm going to pull that slide out, stick it in Cosmics, and then image the whole transcriptome. So I end up with a single slide with 1,200 antibodies and the whole transcriptome. So let's take a look at what that looks like. There's the geomics image on the left. There's the Cosmics image there. And I'm going to zero in on the germinal center of biology. I'm a big fan of immunology. Germinal centers are where it's at. So I'm going to blow up that germinal center and show you what it looks like. It's basically 200 microns worth of a germinal center over there. And I got 30 places where I'm looking at the 1,200 antibodies and how do they change as I go from one end of a germinal center to another. And you can see all those lines. Every line is the signal and noise associated with one of those antibodies in my 1,200 antibody plex. Okay. Then I go over into Cosmics and I look at that thing and I image the whole transcriptome and I look at the biological domains over that germinal center, and that's what you see over there. You see the dark zone, the mantle zone, light stromes, the stromal vascular boundaries, all resolved at single cell. Now I got the protein layer on there, and I go into my large language model and I start harmonizing the data between the protein and the RNA and do that analysis. Take a look at that. Can you see the little those round circles show up? Those are the antibody profile superposed onto the whole transcriptional profile. Okay. Incredible amount of information about what the germinal center is doing there. Each one of them is actually a little different. I could talk about it forever. One of the things we were able to resolve is that it had been hypothesized that the germinal center light zone has got high demand metabolically. A protein unequivocally answers that. That's real data, and it is true. So those people that were hypothesizing that were correct. I think the one thing I'll point out in general is that people say, well, then aren't you just matching up your little mRNA with the protein? And it's a logical thing to say, but when you realize the differences in signal to noise, the signal to noise on the protein signals are 100 to 1,000 times bigger than the mRNA. Okay. So you don't want to sit there and say, how about my little one mRNA or two molecules? And then how does that match the protein? Because the protein signals are so good compared to the mRNA, so strong. So you don't match up an individual protein with an individual mRNA because the protein signals are so much better. But what you can do is look at the pathways that lead to a protein and then equate the pathways with the endpoint proteins, and those do work very, very well. So if you get there, keep that in mind. Okay. And I want to do one more on proteins because this is a great example where you can discover and validate on the same slide. So let's start it off in geomics. Let's take a look at it. We're doing Alzheimer's, neurodegenerative disease. You spend about a week looking at 1,200 antibodies and how they surround an Alzheimer's plaque and how they interact with neurofibrillary tangles. And Miranda Orr discovered more protein biomarkers in a week than had ever been discovered in Alzheimer's because you're doing 1,200 antibodies at a time. She was very happy about that. She found 30 that she wanted to do a large patient cohort and generate a path to the clinic style assay out of that. So that's great. I made the discovery. In geomics, I pulled the slide out, put it in a refrigerator. We took those 30 markers and handed over to my colleague who's here. He's one of the inventors on the CellScape system. So you got two inventors here today. And they said, Oliver, make me a 30-plex panel that's going to image these biomarkers that I just got sequencing barcodes on. Oliver did that in 1 month. So I pull a little slide out of the refrigerator, send it to St. Louis where Oliver's sitting there making measurements. He puts it into the CellScape, and this is what he measured. Let's take a look at these two things. Okay. The left is the geomics image. The right is the CellScape image. Those cells are matched. Every cell here is identical to the one there. Okay. Now, one of the things they were interested in is that little spike between ring 1 and ring 2. That's the green ring and the blue ring. They get a spike and a signal associated with an antibody for phosphorylated tau protein, phosphorylated serine 404, one of her key biomarkers she was interested in. And then boom, look what happens over there. Oliver just imaged that thing. And it's imaged in this little cyan color. So looking at blue ring between the blue circle and the green circle, look up around 2 o'clock, you see two little cells looking at you. They're sort of like, I don't know what you call that, cyan, purple. Those two cells are the cell of origin of the p-tau serine 404 that was discovered over here. We've imaged them and determined the cell of origin of that marker on the same slide. Okay. I discovered something on a slide and validated on the same slide in the same cell. I don't know if you guys have ever fought with nature editors as you try to publish something. They just hammer you to death, right? No one can fight that validation there. It's the same two cells, same slide. So I'm excited about that work, and you can see how you can make that path, discovery in protein land, all the way down to translation. You can do it on the same slide. Okay, so this will be the last thing I talk about. How long does it take to get into the clinic? Geomics now works in Night Diagnostics Lab. Over 82 markers have been clinically validated. They're now at about 110, I believe, 120. And what's it look like? That's a patient report. That's a patient report on geomics as part of a clinical trial that's in breast cancer. 2 arms to the clinical trials, tyrosine kinase inhibition plus immune checkpoint inhibition. So drug companies very, very interested in the phosphorylation status of ERK1 and 2. That's what we measure inside the geomics. Nice sequencing, digital barcodes, quantitative. That patient there has got phospho ERK1 and 2 in her particular core needle biopsy. So that person goes into that arm of the clinical trial work, and we'll see what happens. It was interesting. The pathologist down there called me up and said, I'd love to see that patient imaged. So I said, I'd love to image it too. Let's do it. So he sent me the core needle biopsy. There it is. It's that core needle biopsy. That's from that patient. If you look at our immune profiling panel on Cosmics, the 64-plex, there's phospho ERK1 and 2 in there. So it's already in there. I didn't have to do anything. And then I'm imaging. Phospho ERK is one of those proteins that not activated. It's in the cytosol. It gets phosphorylated, migrates into the nucleus. It's a transcription factor, and that's what turns on all the bad driver behavior out of that tumor cell. Okay, so take a look at that red one down there over on the right-hand side. All the phospho ERK overlaps with DAPI. I color it red. And then you see all that blue stuff that's sort of floating around in the cytosol. That is the non-phosphorylated, non-activated ERK. So unequivocal image that supported the geomics patient report that went to the tumor board. That's nice. I got a repeat core needle biopsy 2 weeks after that patient was treated with that tyrosine kinase inhibitor, and her activated phospho ERK1 and 2 is totally gone. There is not a single nucleus that has activated phospho ERK in it. So they're pretty happy about that. So the drug company knows that that arm, that drug's doing what it's supposed to do. Okay. I'm blinded from the combo one. Who knows? Maybe it reported out at ASCO. I don't actually know. It could be reported out and I don't know about it yet. But I'm blinded from that part of the program, obviously. But unequivocal proof, you can see how this stuff is moving into the clinic. Okay. This is moving into clinical trials. That's just one step away from moving into the clinic. And then this just rounds it out. You have to remember that all the chemistry I do in those microscopes started here. These were all the things I did when I first got the NanoString, was to build investigational use only RNA signatures on barcodes. We were the pivotal phase 3 clinical trial arm of Merck's Keytruda. Probably more money made on that drug than any other drug on the planet. We were the phase 3 pivotal clinical trial work with DLBCL. This is one I did myself, a couple of thousand patients. We got it FDA cleared. That's called PAM50. It's a signature for breast cancer. I think I'm bringing that up is that, if you're doing spatial biology and you're interested in taking it to the clinic, it's worth thinking about the chemistry. Okay. This chemistry migrates into the clinic, can go through the FDA. Okay. So think about how you do your discovery. This is a path where you can make your discovery, make a full-blown diagnostic, and you never change your chemistry. You never have to change it. It's hybridization. It's proven. It works all the way through a regulatory agency. Okay. It's worth thinking about. And I'll just emphasize there's never been a rolling circle diagnostic. Yeah. So if you make your discovery there, you're going to have to convert to something that actually can translate to a clinic, and that can take a long time. Okay. We're not done. I got a brand new signature for antibody drug conjugates. I did this with MD Anderson. It may be make worldwide. I don't know exactly. We're in the process of figuring out how to do regulatory for this thing. There's a ton of incredibly interesting antibody drug conjugates therapeutics out. And we have the highest resolution, reproducible gene expression signature for that that's ever been made. And you can actually find this thing. Oh, it's going to be in clinical cancer research. And maybe it's out now. I don't know. People look at it. And we made this whole thing multiomic, and I won't bore you with all the same things you can do in bulk that you can do in space. So I think this is where I'm going to end. But you can see, a lot of stuff happens in a year. I think when I came here a year ago, I don't think I talked about a single one of those things. So everything that you saw on that screen happened between last July and June the 2nd. This field is really moving fast. It's an exciting time to be doing this. And I hope that, you can do for the patients and the medical care in Korea that we built all these platforms for. Let's solve some diseases and let's actually cure some people, and help them out. That's what the whole thing's about. So I appreciate your attention, and I think I'm handing it off. Thanks.